Hi, everybody. My name is Eddie Nash, and today we'll be talking about our project, Small Molecule Mimetics of Humanin that activate GP130 and normalize neuronal PAKT levels as novel therapeutics for Alzheimer's disease. As mentioned, my name is Eddie Nash, and today I'll be presenting with Sadhana Sankar, Jose Angelo Grajeda, and Liba Faruqi. We're all undergraduate researchers at the Drug Discovery Lab under the guidance of Dr. Varghese John. So to get us started, a little bit about the introduction and the abstract of our project. Alzheimer's disease is a progressive neurodegenerative disorder characterized by the presence of senile plaques composed mainly of amyloid beta peptide and neurofibrillary tangles resulting from the hyperphosphorylation of tau in brain tissue. Alzheimer's disease is the leading cause of dementia, and patients suffer from deficits in cognition, learning, and memory. Humanin is one for acid mitochondrial-derived peptide that has been shown to increase survival of neurons after exposure to Alzheimer's disease insults. The primary extracellular receptor complex involved in its bioactivity is a trimeric complex involving GP130. Humanin binds to this receptor complex as an agonist and activates the downstream PI3K AKT pathway, resulting in increased PAKT levels in neurons. Increased PAKT levels is associated with neuroprotective effects and neuronal survival, and thus Humanin or Humanin mimetic could be highly beneficial in the treatment of Alzheimer's disease. Therefore, a small molecule Humanin mimetic that increases and normalizes brain PAKT levels would be of great value, as it would more likely lead to compounds with brain permeability and allow for probing this mechanism in vivo. We have screened the UCLA compound library and successfully identified small molecule hits with some brain permeability that we hope to optimize for potency, solubility, drug-like properties, and brain permeability through a medicinal chemistry hits lead optimization campaign. Such compounds would be neuroprotective and potentially lead to a new pharmacological class of Alzheimer's disease therapeutic agents.
So next, I'm going to be discussing the methods that we use in our lab to detect compounds that can act as Humanin mimetics. We performed a high-throughput screen of the UCLA compound library and successfully identified small molecule hits that can act as Humanin mimetics. For the high-throughput screen, we developed an ELISA assay to determine the levels of phospho-GP130, as well as an alpha-ELISA assay to assess phospho-AKT levels. As Eddie mentioned before, the protection by Humanin has been shown to be primarily through its interaction and activation of the GP130 receptor, and this interaction results in normalization of the biological target, which is brain phospho-AKT levels. This is why we developed these assays to assess phospho-GP130 and phospho-AKT levels. So the HDS was performed using U87 cells, and these cells were plated at the concentration of 25,000 cells per well in a 96 well plate, and they were treated with 5 micromolar of the compounds in the chemical library for 10 minutes. The cells were then lysed with the appropriate buffer for each analytic and then frozen. Next, as a part of the ELISA to assess phospho-GP130 levels, 100 microliters of the lysis solution was placed into each well and incubated for 2 hours with an anti-phospho-GP130 capture antibody. Then the wells were washed 5 times and incubated for 2 hours with the anti-phosphotyrosine detection antibody. The absorbance was then read at 450 nanometers. As part of the alpha-ELISA assay to assess phospho-AKT levels, 2 microliters of lysis solution was transferred into each well and incubated for 60 minutes with the anti-AKT acceptor antibody. Then anti-SIR 473 was added into the wells and incubated for 30 minutes, after which the signal was read. Next slide, please.
Thank you, Sadhana. So now I'm going to be talking about some of the results from the study. This slide shows sort of the drug discovery process for the small molecule mimetics of Humanin. In figure A, the results from the high throughput screening were that 22 compounds increased PGP130 by at least 20%. That's shown by the solid dots above the dotted line. Likewise, in figure B, the results from the PAKT were that 18 compounds increased PAKT by more than 20%, again shown by the dots above the dotted line. Overall, from the high throughput screening, there were 9 compounds that increased both PGP130 and PAKT by at least 20%, But for more further study, we took the 22 compounds from figure A and selected them for validation. And in figure C, it shows only 4 of those increase both p-GP130 and p-AKT over 20%. Those are the 4 dual hits shown in the box in the top right of figure C. Lastly, we took those 4 compounds, which are C14, C26, P46, and C50, as well as DDL812, peptide 3, and 1 new chemical entity. And we tested them at 0.5 and 0.05 micromolar concentration, and then assessed p-GP130 levels. This is figure D. From this experiment, we found that only C14, C46, DDL812, peptide 3, and the 1 new chemical entity increased p-GP130 over 50% at 0.5 micromolars. That's shown by the bar graphs above the dotted line in figure D, which is what we're looking for. And next, I'm going to pass it on to Liba to talk about the next steps and our conclusions.
Yes, so as shown in figure D, we did find that of the compounds tested, there were 4 dual hits, C14, C46, DDL812, and peptide 3, and all increased phospho-GP130 signaling by over 50%. And so with these results, we were able to conclude that we should optimize the screening for phospho-AKT signaling, and specifically with anti-SER473 signaling. We are currently working on optimizing it as an alpha ELISA assay to screen the hits. Thus, we will also be conducting further dose response testing for the compounds that increase phospho-GP130 signaling at various concentrations and developing optimized analogs. We will be running more ELISA assays to obtain EC50 on the hit analogs and quantify the phospho-GP130 signal. EC50 is the half maximal effective concentration, which is a measure of the concentration of a drug that induces a response halfway between the baseline and maximum after a specified exposure time. Thus, we will be running these ELISA assays to obtain the EC50 on these analogs and to quantify the signal. I will be passing it on over to Sadhana to discuss our acknowledgements.
Thank you, Liba. So to conclude our presentation today, we would like to express our sincere gratitude to Dr. Varghese John, our PI, as well as Mr. Jesus Campagna, our lab manager, and Dongwook Wee, the Humanin project leader. They have provided direct guidance for us throughout this process, and we really appreciate their assistance. Thank you so much, everyone, and I hope you enjoyed our presentation.